Embryology, which is the study of young ones, is the focus of Chapter 3, and we will be looking at developmental changes that occur prior to birth. And so with human development, we are looking at two things. We're looking at differentiation, to look at the formation and organization of the diverse cell types, and then reproduction to ensure that new individuals are produced from generation to generation. Embryogenesis is collectively known as the developmental process that occurs for humans. And there are three different stages that occur. The first is the pre-embryonic period, and this is the first 2 weeks of development right after fertilization of the egg, and where we have single cells of the sperm and the egg that become spherical, and then they start forming a blastocyst. And this is done through mitosis and by cleavage of this blastocyst. And once this blastocyst, which is going to be a bunch of cells, are going to implant into the lining of the uterus. Then, after that second week and the implantation into the uterus, we enter the embryonic period, which this is where we have the term the embryo. And this period lasts from weeks 3 through 8. And here we start seeing major organ systems that start to appear. And this is done through gastrulation, where the blastocyst starts to form 3 different layers of differentiation into its specific organ and organ systems. And then the rest of the duration in utero is known as the fetal period, and thus we have the term the fetus for the human being. And this lasts either 38 weeks, but can go into 39 or even 40 weeks, having a total gestation of 42 weeks, and that is absolutely normal. Here we have continuation of growth of the fetus as well as complexity of all of the organs. And these complexities are going to be determined by organogenesis, where we have those germ layers are going to derive and give rise to all the different organs within the body. Right after birth, we humans spend a lot of time undergoing growth and maturation. And so the body will continue to grow and then develop, but also we have our sex organs start to become more mature. The sex organs, which are ovaries in females and testes in males, start to produce cells or sex cells or gametes, and these then start the process of gametogenesis. Gametogenesis is important because this is the reproductive phase for development to help with the continuation of our species. And our hereditary information is carried on our chromosomes, which, if you recall, we have 23 pairs of chromosomes where 22 pairs are autosomes, and these are have information that pertain to most of our characteristics, like our skin color, our eye color, hair, and all that fun stuff. And then we have one pair of sex chromosomes, and this is where you'll be determined if you are considered to be a female. So if someone has 2 X chromosomes, or male, which has 1 X chromosome and 1 Y chromosome. Now there are some alterations that occur during meiosis where you can have either 1 sex chromosome, so you can have only an X or only a Y, or you can have 3. And so these are just due to, again, changes with meiosis. And so one member of each pair of your chromosomes is inherited from each parent. So you have 23 chromosomes that came from your mother and 23 chromosomes that came from your father. So together you have 46. However, there are only 23 pairs altogether. So a cell that has 23 pairs is considered to be diploid. Gametes, on the other hand, are considered to be haploid, where they only contain 1 of the pairs. So only a total of 23 chromosomes instead of 23 pairs. So gametogenesis begins with cell division or meiosis, and the sex cells produced in the female are going to be the secondary oocytes, and the sex cells produced by the male are going to be sperm. And so now let's look into meiosis a little more. Before meiosis can occur, we have interphase, which is part of the cell. And here, during interphase, we have the DNA in each chromosome becomes replicated or duplicated in the parent cell. These replicated chromosomes are composed of two identical structures known as sister chromatids. Each sister chromatid contains an identical copy of DNA. The chromatids are attached to a specialized region of the centromere. The chromosomes composed of sister chromatids resemble the letter X, and it's not the same as a pair of chromosomes, which you would have two X's. Thus, after interphase, there are 23 pairs of replicated chromosomes, and once this DNA replication has occurred in interphase, then we can enter meiosis. With meiosis, we have the sex cell division that starts with the diploid parent cell and produces haploid daughter cells that are our gametes. Meiosis produces 4 daughter cells that are genetically different from the parent cell, and we have meiosis produces a daughter cell that are haploid, which we have crossing over that occurs, which is the exchange of genetic material between the two gametes. And thus, this helps to shuffle our genetic code. And that one example would be is if someone's ever said to you, oh, you have the eyes of your mother, but then the cheeks of your father. Well, that would be due to crossing over during meiosis, where you've inherited from both parents. So meiosis begins with a diploid parent cell that's located in the gonad, which is the ovary or the testes. And within the cell, 23 chromosomes came from the organism's mother, or the maternal chromosomes, and then 23 came from the father, or 23 paternal chromosomes. The first stage of meiosis, or meiosis 1, is known as the reduction division, and this is when we have the homologous pairs of chromosomes are separated when the cell divides. The result is 2 cells, each of which contain 23 chromosomes that consist of duplicated chromatids that are held together at the centromere. And so since they split, this is where we have the term, the reduction division. Meiosis 1 begins with prophase 1, and this is where homologous replicated chromosomes in the parent cell pairs up to form a tetrad, or tetra for the number 4. The process by which the homologous chromosomes pair up is called asynapsis. Here we begin the crossing over of the maternal chromosomes as well as the paternal chromosomes. And this also thus allows for that genetic diversity to occur with each generation. This prophase 1 of meiosis 1 ends with the breakdown of the nuclear envelope of the cell. Following prophase 1, we enter metaphase 1, where we have the homologous pairs of each tetrad that lines up on either side of the midline or the equator of the cell, and it forms a double line of chromosomes. There's a random alignment of the chromosomes in terms of the maternal and the paternal chromosomes, so that it's not that all of the maternal are going to line up on the left side and all the paternal are going to line up on the right side. So this is just completely random. And so because of this, we have this independent assortment. And When we have the spindle fibers then formed by the microtubules that extend from the centrioles at opposite ends, then we have this attachment of the centromere to each homologous replicated chromosome where we're going to start to have that separation of the cell. Following metaphase, then we enter anaphase 1, and this is where the homologous pairs of chromosomes separate and are pulled at opposite ends of the cell. And this is based on that division in metaphase of the equator. And so the chromosome pairs begin to separate, and this is known as the reduction division because each daughter cell only receives 1/2 of the starting number of chromosomes. Thus, there are only 23 chromosomes instead of the original 23 pairs of chromosomes before. And so each pair of chromosomes are no longer together. However, each sister chromatid are still matched together. Following anaphase 1, we have telophase 1 and cytokinesis. Here now, we have the chromosomes that have been replicated are now at opposite ends of the cell. Then there is a nuclear membrane that may reform around these chromosomes. And so a cleavage furrow will form and have the cytoplasm of the cells divide. Hence the term cytokinesis, where now each daughter cell has its own cytoplasm to produce, now it has 2 new cells. Each daughter cell has 23 replicated chromosomes, no longer the 23 pairs, so the reduction division during meiosis. And now each replicated chromosome, though, still has 2 sister chromatids that are bound together. So then after telophase 1 and cytokinesis, then there's a round of meiosis 2 to now separate those sister chromatids of each chromosome. Now we enter into meiosis 2, which is now the separation of the sister chromatids, where prophase 2 is going to start the event of meiosis 2, and each of the 2 new cells now will have the nuclear envelope start to break down, and the chromosomes will collect together. Now the only difference between meiosis 2 and or prophase 2 and prophase 1 is that there is no crossing over that occurs because the chromosome pairs are not present. Following prophase 2, we enter metaphase 2, and here we again have the spindle fibers that are going to extend from the centrioles to the centromere of each chromatid, and then the chromosomes, which are of the sister chromatids, will now align to form a single line along the equator in the middle of the cell, very similar to that of metaphase 1. Following metaphase 2, we enter anaphase 2, where we have the sister chromatids of each replicated chromosome are pulled apart at the centromere and become separated. Now, each sister chromatid now is termed a single chromosome, and it is pulled to the opposite pole of each cell. And the last stages of meiosis 2 are telophase 2 and cytokinesis, where the single chromosome arrives at the opposite end of the cell. There's a reforming of the nuclear membrane around each cell, and then a cleavage furrow forms where the cytoplasm then divides into both cells as well. So this end process of meiosis occurs, and we have now 4 daughter cells from the original single cell, and these daughter cells are haploid because they only have 23 chromosomes and not 23 pairs of chromosomes. These daughter cells then will mature into secondary oocytes in females, and they'll mature into sperm in males. In females, the sex cell or the stem cell that produce the oocytes are the oogonia. And so here, I'll circle these first at the top. and these are found within the ovaries. In a female fetus, all of the oogonia start the process of meiosis and form primary oocytes. And so here's the next thing that I circled. Primary oocytes stay in prophase 1 until the female reaches puberty. And so each month, a number of primary oocytes begin to mature, and usually only one of them becomes a secondary oocyte. So here's the next thing I circled. When the primary oocyte completes the first meiotic division of prophase 1, metaphase 1, anaphase 1, and telophase 1, 2 cells are produced. However, there's an unequal division of the cytoplasm between the two, and so the secondary oocyte receives a lot more cytoplasm than the first polar body does. The polar body is just a non-functional cell that will eventually disintegrate within the female body. Only the secondary oocyte that has the potential to be fertilized, the first polar body does not. So the secondary oocyte then can be ovulated, which is expelled from the ovary into the uterine tube, again, once the female reaches puberty. So if the secondary oocyte is not fertilized, then this degenerates about 24 to 48 hours after ovulation, and the secondary oocyte still remains in metaphase 2. Now, if the secondary oocyte is fertilized, then it finishes the process of meiosis. Then we get 2 new cells that are produced, and the divisions of the cytoplasms are also unequal. The cell that receives little cytoplasm becomes another polar body, which is here, the second polar body, which too will just eventually disintegrate just like the first polar body. However, the cell that receives the majority of the cytoplasm is called the ovum. And down here, ovum. And the ovum has the nucleus that combines with the sperm to produce the diploid fertilized cell, or the zygote. Now, typically, we only have one secondary oocyte that is expelled, and so there's one fertilization of an ovum. However, there can be times where, say, one side of the uterus, but rather the ovaries, releases 1 or 2 or sometimes 3 ova, and so we can get multiple successful pregnancies because of that. So that's when you get twins and triplets and so on and so forth. With that, that are produced are called sperm or spermatozoon. And the process of sperm development is called spermatogenesis. The parent of one or stem cell that produces sperm are called spermatogonia. And so here, I'll circle that first, or spermatogonium, which would be the singular of that. Spermatogonia are diploid cells that reside in the male gonads of the testes. Each spermatogonia will first divide by mitosis to make an exact copy of itself. And this is called now the primary spermatocyte. Each primary spermatocyte then will divide into 2 secondary spermatocytes, and each secondary spermatocyte will divide into 2 spermatotids. Spermatotids contain only 23 chromosomes. They must still go along to create sperm. And so the process is known as spermiogenesis, where the spermatids lose much of their cytoplasm, and then they grow this long extension or long tail called the flagellum. And the newly formed sperm are haploid cells that exhibit a distinctive head, a midpiece, and a tail. Thus, from a single spermatocyte, we have 4 new sperm are formed. During the pre-embryonic period of development, we first begin with fertilization, where we have the 2 sex chromosome or cells fusing to form a new cell that has the genetic material from both parents. And so the big thing with fertilization is that now the cells have a diploid number of chromosomes. Also at fertilization, the number or the sex of the organism will be determined here, and then also cleavage of the cell will begin. Fertilization occurs within the largest or the widest part of the reproductive tract within the female, which is found in the ampullae, and we'll cover more of that in the reproductive system later in the semester. But interestingly, so the secondary oocyte remains viable in the female reproductive tract for no more than 24 hours. However, male sperm can be viable for up to 3 to 4 days following ejaculation. Before the sperm can be viable and be successful in terms of fertilization, what has to occur is this period of conditioning, which is known as capacitation. During capacitation, a glycoprotein coat and some proteins become removed from the sperm plasma membrane, this allows the sperm to respond to the different signals that are found within the oocyte, and then also in order to find where the oocyte actually is within the female reproductive system. The acrosome, which is the tip or the head of the sperm, also has to have these digestive enzymes that can break down the protective layers around the secondary oocyte. And the enzymes that are released when the sperm comes into contact with the secondary oocyte so that it can easily penetrate through the different layers of the oocyte. So, interestingly, there are millions of sperm that are deposited within the vagina and then into the female reproductive tract during intercourse. However, only a few hundred actually reach the oocyte in the uterine tube, and then only the first sperm to enter the secondary oocyte is able to fertilize. Once that does occur, then the remaining sperm are just prevented from penetrating the oocyte. And then the sparks fly, like in the video that you will watch in a little bit. After fertilization, the zygote begins a process where it becomes more and more cellularized. And so it keeps dividing from the 2 cells, and it'll go to 4 cells and 6 cells, or 8 cells, and keep doubling in number of cells. And so once we go past the 2 cell stage, then this is known as cleavage. This increases the cell number, but it does not increase the cell size just yet. The size will stay the same until it implants into the uterine wall of the mother, where it can provide additional nourishment and increase in size there. So there are different terms that are used for the number of cells that are observed. So once we have a 16-cell stage, this is called the morula. And once we pass the morula stage, then there is a compaction of all these cells, and so we enter the blastocyst stage. And so within the blastocyst stage, we have 2 different components that occur. The trophoblast, which creates the outer ring of cells, and that has a fluid-filled cavity and will form the chorion later on. And then also the embryoblast, or the inner cell mass, which will be within one side of the blastocyst. And this is important because this is where the cells become pluripotent and means the power to differentiate into any cell or tissue type within the body. By the end of the first week after fertilization, the blastocyst enters the lumen of the uterus, and then implantation occurs where the blastocyst burrows into and embeds within the endometrium of the uterus. So this occurs at about day 7 following fertilization, and by day 9, the blastocyst will have completely burrowed into the uterine wall. And so it makes contacts then with the pools of nutrients within the uterine glands that supply the developing organism. And that's why this implantation begins during the first week of development but isn't complete until the second week. Also, during the implantation phase, we have this hormone called the human chorionic gonadotropin, or HCG hormone, that signals the female reproductive system has been fertilized successfully and implantation has occurred. And so this is one, when you go to the nurse or doctor's office, this is the hormone that they test for in order to confirm if somebody is pregnant or not. And following the next 3 months after implantation, also there will be large amounts of estrogen and progesterone that are going to be expressed with the hormones, and this is to help build the uterine lining in preparation for the amount of growth that will occur for the fetus. Starting at about day 8 of the pre-embryonic period, where implantation has occurred, there are extra embryonic membranes that form, and these are to assist the embryo in vital functions like nutrition, gas exchange, and removal and storage of waste materials as it develops. So one of the first extra embryonic membranes is the yolk sac, and this is the first one to develop. Unlike with reptiles and birds, humans do not store yolk, but it is an important site for early blood cell and blood vessel formation. The next, the amnion, is a thin membrane that eventually closes the entire embryo in a fluid-filled sac called the amniotic cavity. And the purpose of this is to protect the embryo from drying out or from desiccation. And this membrane also secretes amniotic fluid that bathes the embryo throughout the entire duration of its development in utero. And the last extra embryonic membrane is the chorion, which is the outermost outermost membrane. And the cells within the functional layer of the endometrium of the uterus will eventually form the placenta, and this is where the site of gas exchange or of exchange between the embryo and the mother occur. The connection between the embryo or fetus and the mother is via the richly vascular placenta. So the main functions of the placenta are to exchange nutrients, waste products, and respiratory gases between the maternal and fetal bloodstreams, to transmit maternal antibodies from the immune system to help target against bacteria and viruses for the developing embryo or fetus, and then also to produce hormones, primarily estrogen and progesterone, to maintain and build the uterine lining to help again maintain and prepare for development of the embryo and fetus. So the placenta begins to form during the second week of development, and it develops from the chorion, while the maternal portion of the placenta forms from the functional layer of the uterus. And the early organism is connected to the placenta through a connecting stalk. And so this connecting stalk contains the umbilical arteries and veins later on, and will later become the umbilical cord. The finger-like structures of the chorionic villi form from the chorion, and so these chorionic villi contain branches of umbilical vesicles, and adjacent to these are the endometrium, which contains the maternal blood. So interestingly, the fetal blood and the maternal blood do not mix. However, their bloodstreams are so close to one another that they are able to exchange different gases, waste products, and nutrients from the two. Also, though, undesirable substances like drugs, alcohol, and nicotine can also cross that placental barrier and can cause damage. And this is true even for over-the-counter medication. So, for instance, aspirin only takes 13 seconds from ingestion to crossing the placenta and to get into that fetus. So, it is very fast in terms of the delivery of that content for that embryo or fetus. Entering the embryonic period, we have the establishment of three primary germ layers through the process of gastrulation. And so these three layers then form the specific tissues and organs that we find throughout the body, and this is called organogenesis. By the end of the embryonic period, which is about at week 8, the main organ systems have been established, and the major features of the external body form are recognizable. And so sometimes women might have to have an early ultrasound, and through the ultrasound, you can detect all the different organ systems and through it. So the first stage that we have is gastrulation, and this occurs during the 3rd week of development, which is immediately after implantation. And this is probably the most critical period in the development of the embryo because we have 3 primary germ layers that occur and form that, again, help to make the different tissues developed. So we have the ectoderm, which is the outside layer. We have the mesoderm, which is going to be the middle layer, and then the endoderm, which will be the inside layer. So, for example, the ectoderm is going to form part of the nerve or the epidermis, part of the nervous system, and, for instance, part of the sensory systems. The mesoderm, the middle layer, will help to form the muscle tissue, our connective tissue, also part of our heart and kidneys. And then, last but not least, the endoderm, which is the inside germ layer, will help to form the liver, the respiratory tract, the gastrointestinal tract, and part of the digestive tract as well. Once these 3 structures or these layers have formed, then the correct term is that this is now an embryo. And so gastrulation begins with the formation of the primitive streak, and this is a thin depression that's on the surface of the epiblast. And there a head region or cephalic region of the streak, and this is known as the primitive node. And then there also is a elevated area that is called the primitive pit. And from these, we have the formation of the different germ layers due to the movement of cells that are constantly being invaginated. During gastrulation, so a 3-week embryo resembles a flattened disc-shaped structure known as the embryonic disc. So after the 3 weeks, then we have 2 types of folding that occur for the embryonic discs. And so the first one is the cephalocaudal folding, and this occurs in the cephalic and the caudal regions of the embryo. This helps to create the future head and the buttocks region for the embryo, where we have the embryonic disc and the amnion grow very quickly. However, the yolk sac does not grow at all. And so this differentiation causes the head and tail regions to fold on themselves. The second type of folding that occurs for the embryonic disc is the transverse folding that occurs on the left and right side of the embryo curve and migrates towards the midline. As the sides come together, they restrict and pinch off the yolk sac. Eventually, the embryonic discs fuse in the midline and creates a cylindrical embryo. So now this allows for the ectoderm to now be on only the exterior of the embryo, and the endoderm to be confined to the internal region of the embryo, and then the mesoderm will then fall in between the two. And so the transverse folding helps to create a cylindrical trunk or dorsal region for the embryo. After the embryo undergoes the caudal or cephalocaudal and transverse folding, then the ectoderm is located on the exterior surface of the embryo. And so the ectoderm is responsible for forming the nervous system tissue and the epidermis of the skin and epidural derivatives such as hair and nails. The mesoderm is divided into 5 different categories. And so the first one is that it is responsible for packing cells tightly along the midline. And then this helps to form the notochord, which is the basis for the central body axis and the axial skeleton, and helps to form the neural tube. Also, we have the paraaxial mesoderm, which is found on both sides of the neural tube, and this forms the somites, which are the block-like masses responsible for the formation of the axial skeleton, most of the muscles, and most of the cartilage, dermis, and connective tissue of the body. Lateral, we have to the paraaxial mesoderm are cords of the intermediate mesoderm, and so these are responsible for the urinary system and reproductive system. Fourth, we have on both sides are the lateral plate mesoderm, and these give rise to the cardiovascular system, the lining of the body cavities, the thoracic and abdominal wall, and the connective tissue components of the limbs. And then lastly, we have the head mesenchyme, which forms the connective tissue and musculature of the face. The endoderm becomes the innermost tissue when the embryo undergoes transverse folding, and so it forms the structures and the linings of the digestive, respiratory, and urinary tracts, as well as the thyroid gland, parathyroid gland, thymus, and portions of the tonsils, as well as the liver, gallbladder, and the pancreas for us. Once the 3 primary germ layers have formed and the embryo has undergone the cephalocaudal and transverse folding, then organogenesis begins, and this is the development of organs. And so the upper and lower limbs attain their adult shape, and there's the rudimentary outline of all the organ systems that have been developed by week 8. At the end of the embryonic period, the embryo is no longer than 2.5 centimeters, which is only about an inch, and it already has the appearance of a human. However, during this embryonic period, the embryo is very sensitive to tetragens, which are substances that are known to cause birth defects. And so these include alcohol, tobacco smoke, drugs, viruses, medication, so even over-the-counter aspirin, ibuprofen can affect the embryo. And so this is a time when it's very vulnerable, and thus a lot of women might miscarry, and the chances are at about 33% of it happening during this embryonic stage of development for that embryo. So the last stage of development is the fetal period, and this begins at week 9 and lasts until birth. And here we have maturation and growth of all the tissues and organs, since the organ systems have already been established. And so what will occur are now commonly known as the different trimesters. So as you exit the first trimester, which is about weeks 1 through 12 or 1 to 13, then you enter the second trimester, which ends at about week 24, 25, and then the third trimester, which will be through birth. And so here we'll have the bones starting to ossify, we'll have the hair starting to grow all over, and also the fetus will adapt to different tastes from the mother, start being able to recognize different sounds that are common. And so like a fetus can recognize a certain voice or a song that's played a lot, or even a dog barking. And so with it, the head size also increases very rapidly throughout the gestational period.